You talked about how the FLC Jane gets switched from on to off. Can it be switched on again so that the Arabidopsis can live another year? Or is it that Arabidopsis can only live for 1 year?
Arabidopsis thaliana only lives for 1 year, but actually a close relative, which doesn't matter what the name is, switches it back on so that it can live several years. It's a perennial. So
have you looked
absolutely.
into how it gets switched back on? Is it similar?
Yeah. Well, we think, do you remember I showed you this northern Swedish type that if you don't give it enough cold, it switches back on? It's something to do with a mechanism like that. There are some changes in the sequence which just mean it's a little less stable, and so therefore it'll switch back on.
Great. Next question, please.
I've got a flowering cherry in my garden that I was inspecting just this afternoon, and it appears to be completely dead. But if you break off a twig, you can see it's green, and
Yeah.
it hasn't.
So it didn't
produced
flower
leaf,
this
hasn't
year
produced
at
flower.
all?
No, not at all. And yet it appears to be still alive.
Ah.
Is that? I mean, I'm guessing this is a sudden heat surge or cool surge or something like that that's affected it.
Yes, I'm not a cherry expert, I have to say. Did it have any leaves? I mean, just nothing happened?
Nothing at all.
Nothing
it
at all.
looks,
So
it
it's
looks
pretty
like it's
catastrophic.
midwinter, and
I
yet
think
if you-
it's probably been very badly either drought stressed, and it's just decided to give up for this year. But it's still kind of little bits in there. I mean, I think there was drought will do that, and it will tend to sort of miss a year completely. If you're lucky enough, some bits of it will come back, but probably not all of it.
There's some, sorry to add one more, there's some apples, I know you're not an apple expert, there's some apple trees as well, and I've noticed that they'll quite often skip a year, they won't
Yes,
flower one
well,
year.
some trees skip 7 years, some trees, and actually very interestingly, some trees know not to have nice even flowering because the pests, if they flower evenly, adapt to knowing that evenness is coming. So they basically randomize it to sort of out-compete the pests. So there's all sorts of strategies out there. But drought, I think, and very, very severe, we didn't have a very severe winter last year, so I would think it's probably drought that's hit your cherry and your...
Well, I do love the science in Gardener's Question Time, so here we are. We're the Royal Institution.
I always dread the Gardener's Question Time question.
We need to get you there, because this is where the answers are in science. Next question, please. Thank you, at the front.
I was just wondering about the
Arabidopsis.
Yeah. In countries which don't have such a cold winter.
Yes.
So presumably they wouldn't have be cold enough even for the short enough time to be
That's right.
switched on. So how does the mechanism work, or do they not flower?
So those are the ones which have this rapid cycling strategy. Do you remember there were these two strategies, and depending on the environment, one or other is more reproductively successful. So in the south, they tend to be the rapid cycling type, so they don't need winter at all.
There was a question in the middle as well, sorry.
This is adding to the gentleman's question. I have 3 very mature apple trees, which for 42 years I've always flowered profusely. This year, the number of flowers could be counted on one hand.
Yes.
Drought?
Well, I do listen to Gardener's Question Time, and this rings a bell. And I sort of remember there are lots of these. The last couple of years have been fairly extreme in terms of shortage of water and quite extreme temperature fluctuations. So I would think it's one of those two. But I think they would say, look after it and it might come back.
Oh,
but
yes, I'm going to look after it. They're far too important.
We have a question up here, please. Thank you very much.
Hi. In the Arabidopsis, in Edinburgh and
Yes.
Sweden. So were they genetically identical? And so is it just a protein? So that protein which is turning it.
Yes.
So that protein requires 4 weeks, and the other
Yes.
protein in Sweden
Yes.
requires. So otherwise, it's
Otherwise,
genetically
it's
the
the
same.
same. So it's the gene that is the thing that's slightly different, the FLC gene between those two types. But you can make something that is identical genetically, apart from that gene, with the 4 changes, and that's enough to go from 4 weeks to 12 weeks. So it really is just those 4 small changes that make that difference.
We're going to take a question from the balcony, please.
Hi, it was very interesting in the talk to hear about epigenetics and how your research is helping us understand more about that. I guess I kind of understand about DNA reproduction, how the bases get copied. Could you say a bit more about how the epigenetic state
Yes.
gets copied? Because you said that the cuttings preserve that through cell division.
Yes. Very good question. So DNA methylation, which I didn't talk about, we know more about how that's copied. So the 2 strands of DNA both contain a methylation on a particular base. And actually, when DNA is replicated, of course, each copy then gets a new copy put down, so that you would have, of the new copy, only 1 strand would have the DNA methyl base. But what the enzyme does is it recognizes this strand and it adds it to the other strand in the same position. So there's a reasonable understanding for how you copy the methylation mark if it's a DNA methylation. For those protein complexes, the nucleosomes, it's harder to understand because through DNA replication, as the DNA helix unwinds, so you have the 2 strands and each strand is then copied, all of those proteins are stripped off during DNA replication. So somehow you haven't got a template anymore to copy. So there's a lot of research going on to understand, so how are the proteins, the right color modifications, put back in the right place? And the one possibility is that you hold, so that when they strip the proteins off, they don't just sort of float around the whole cell or the nucleus. They're held nearby, so that as the DNA is replicated, they're then put back on again. But of course, you only have half as much as you need, so you then have to fill in. But it's a much more complicated process how to copy that. And it's still a very active area of research in a lot of labs. So I don't really know the mechanism yet, but it's very reliable. And the other thing that's quite surprising, I just talked about this gene. If you look along the chromosome, those red modifications are very clear over the gene, but they have very distinct boundaries. So beyond the gene, then it's not red anymore. So somehow, whatever it's copying has got a very defined boundary, and those are very precisely maintained. And so again, no one really knows how that works yet.
Any more questions? The gentleman here, please.
With the change in winter, it could be there's not enough cold for
Yes.
vernalisation.
Yes.
Therefore, they wouldn't flower the next year. Can they move to quick cycling, multi-cycling, or quick cycling, in fact,
Yes.
rapidly enough to survive?
Well, that's, I think, really the interesting question is how fast can these things change? It would require a base mutation. So do you remember the Frigida gene that actually had a mutation in it? Now, one mutation can arise, so it's sort of based on the mutation rate. It's not that frequent, but as soon as it arises, it will then be reproductively very successful, so it will be selected very strongly. So in an evolutionary sense, one mutation will happen very quickly. It won't necessarily happen in 1 year, but in a short evolutionary time, it could do 10 years maybe.
So
They wouldn't switch backwards and forwards from 1 year to the next, though.
So I do have a question related to this, which is, given the forecast changes in temperature, by the end of the century, and the bandwidth that we're looking at in terms of those potential error bars, given the work that you're doing, and actually the whole of the John Inner Centre, in terms of what that means for the future of agriculture, are there forecasts regarding what that means regarding the world and food sources?
Well, I go back to this slide because actually this is a sort of schematic of a mathematical model that if you take all the temperature inputs and put it in, it will predict when plants will flower. So now we're starting to take forecasts and models to say, okay, what will actually, because it's not intuitive in terms of a 3-degree increase and exactly what it's going to do because of the complexity of the temperature sensing. But now we can actually really predict what's going to happen. So,
and in basic terms, I assume it's not very pretty, but how not pretty is it?
Well, surprisingly, vernalisation per se is not going to be affected that much because these things are vernalised in the field by Christmas, and yet they still sit there until April. So it's not totally terrible at this point in time.
How incredibly heartening. Any more questions, please? Yes, in the middle. Thank you.
Thank you. You mentioned that they might vary these things because of the periodic attacks by insects and pests.
Yeah.
There's another side of that, and I'm an electronics engineer. I don't know anything about plants, but I do know a bit about prime numbers and Fibonacci numbers. And there's something called a cicada, which comes out in all sorts of very strange periodic time, every 17 years, every 13 years. They're usually curious numbers, and that's the way it seems the attacking insects adjust their life to get back to attack the plants. I wondered if you take this into account. Does it apply in anything in this country or only in USA?
Yes, I mean, Arabidopsis is somewhat boring in this respect in that it just flowers every year, so we haven't really done that. A colleague of mine in Japan was looking at beech trees, which do this masting, as it's called, this sort of intermediate flowering years. There's very little work on it, surprisingly. I watch Brian Cox, The Planets. I'm sure you're watching that at the moment, and it's amazing to me what people can do, send up satellites, and 7 years later they land in exactly the right place at the right time. And then I look at biology and I think, how come we don't understand more? And all sorts of questions, very nice questions to tackle now, and the molecular information we have now is fantastic. So it's a really great time to come into science because you've got all the tools to ask all sorts of interesting questions. So it's actually just identifying the question that's the key.
Fantastic. We have another question in the balcony.
If due to climate change or something, winter wasn't to happen, would the entire species die out?
So Arabidopsis, let's take that for a starting point, has a lot of seed in the soil. There's a seed bank. And only a small proportion of those come out every year. So let's say there was a terrible catastrophe for 1 year. Well, there's a lot of stuff in the seed bank, and that's true for most native plants, that would regenerate the population. It's not true for crops, of course. If you've got a bad winter and you've put your crops out and crops fail, well, we're really in trouble. And I think, we do not have a very good food security at the moment. But in terms of natural plants, I think there is an element of buffering. Plants, as I showed you, different winters are really quite different. They have to adapt to different things. And sometimes if it's a bad winter, the cherry and the drought, it sort of doesn't do very well, but it can come back. So I think, it's not going to completely wipe things out one bad winter. Several in a row, and then I think you've got a problem.
Right. Another question over here, please, Martin. Thank you.
Thank you. One thing struck me that you said. You said that the DNA in this sequence is the same in mammals and humans. Presumably, as far as we're concerned, this particular bit is dead code. But what you're saying is that the same sort of processes and mechanisms might apply to other parts of genetic sequences, and that might be the most important thing of the whole lot.
Yeah, so when I said the sequence, it's every gene is made up of a sequence of bases, And 50% of those genes, you could line them up and say, okay, that's the human gene and that's the Arabidopsis gene. And they wouldn't be exactly the same, but they would be similar enough that you could take it and put it in the other organism and it would work. So, you know, same, but with changes that have occurred during evolution. That I think it's quite remarkable. And that's why 50% of plant genes are, you know, for basic stuff in cells is the same in humans and plants. So that's why I think, you know, there's a lot to get out of plant biology for these basic processes. We don't all have to work in humans necessarily to understand what they do. Yeah.
Any more questions? Yes, again, of course.
I've already asked a question, but I thought I'd take the opportunity. FLC, the way that you had it working where it either turns off and turns on, would you say it's analogous to like quantum physics, where you have enough energy, it can move up a shell, and then once you lose that energy, it moves back down to that. So is that how it works? Like,
well, it's not really an energy state, but it's, you know, opposing modification. So they're mutually exclusive. So Martin likes to call it digital. So it's more like a computer bit. You know, you're either one or the other, but you can't be both at the same time. And that gives, I think this is going to be true in biology in a lot, where you want to switch something. Having these opposing states, which are in a bit of a tug of war with each other, means you're either one or the other. And flipping backwards and forwards is quite a common network in biology. So it's not so much going up to a high energy state and then falling back. It's more just two opposing states battling it out.
Right.
Sorry. So is it almost, I mean, you've got factors that affect it, but it's almost random, like how it can be.
Well, so then the question is, what is required to switch you from green to red? So there are things that target the complexes too. They're protein complexes that deliver those modifications. And there are such things called non-coding RNAs. So in fact, you know, the genome sequencing has shown us that so much more is made than we thought. We thought only the genes were sort of made into RNA and made into proteins. But now actually, if you look hard enough, every bit of DNA is made into RNA, and a lot of that is involved in targeting of these chromatin machinery.
You said that the change in temperature, change in day length, and hormones all target the same protein.
Yes.
So that in the case of the plant you are working with, it's the temperature that...
Is that?
The temperature is what makes the difference.
Yes.
So are there plants where the day length is the important factors and other plant is hormones, and will those plants be totally independent of, say, if it's one that is affected by daylight, will it not be dependent on temperature at all, or is there a combination?
No, very, very good question. So if you take something that is very strongly photoperiodic, and then you knock out that photoperiod pathway, suddenly you reveal a sensitivity to vernalisation or hormones that you hadn't seen before. So they're all there, it's just one is dominant, and that's what you see. But when you take it away, you now reveal all the rest. So it does look like they're all there, and it's just the predominance which is changing.
Super. We have a question at the front. Thank you.
If temperature is the predominant mechanism for the vernalisation, in the tropics, for example, when we visited Costa Rica, some plants which suffer very little temperature variation were still showing seasonality in their flowering.
Mm.
Have you looked at how that can be happening?
Well, I'd love to be funded to go and do something in Costa Rica. That's first point. I think, but very little real understanding of that what happens in the tropics is that it's night temperature that plays a key role. So the dip in the night, so it's more that sort of rather than a longer term temperature, that the plants, let's say in the northern latitudes, would be monitoring. They follow night dipping. And orchids, I think, are the same. It's the night dip in temperature which they're monitoring. So there is a seasonality, but it's just a slightly different monitoring.
In that case, could it be a kinetic effect that the quantity of the inhibitor that is being produced that wraps the gene and inhibits its action, that is perhaps unstable in temperature? It's generated continuously at a low temperature, and as it warms up, it breaks up, and that it's governed by the quantity of that that's present in the thing? Is that possible?
Absolutely possible. There's a lot of very unusual mechanisms, actually. There's one that we've discovered, not just like that, but totally unexpected, in that, do you remember I showed you the protein that went up very slowly in the cold? Well, we were trying to work out how can it go up so slowly, because actually if you, in a normal biochemistry, if something's turning up, the shape of the graph is that it's sort of like that. It's a classic production increase. But this particular one goes up in a very different way. It goes up very, very slowly. And when we mine into that, it turns out it's a very, very stable protein. And it's the dilution by the cells dividing that influence its accumulation. And in the cold, the cells don't divide so much. So now it can go up slowly because it's not being diluted, which is sort of opposite to what you know. But that's a mechanism which was totally unexpected as a way of perceiving temperature.
So if there are any funders looking
Costa Rica,
for research opportunities, there are two things, research opportunities. I know that there will be sort of an interesting project there. But Caroline, I would also be interested to know what the mix of the time between you spending research and when you go versus your students, because it may be rather different from the mix we saw with Sweden.
Yes.
Right, any more questions? One at the back, please.
My question's in relation to agriculture. So if you were to take out drought and bad flooding, and you look at harvests year on year, you still get quite variations in yields. So is that yield difference dependent on the FLC gene or how long it's switched on and switched off? Is there a direct correlation there between yield outputs?
Right. Now, most yield outputs would be probably in plants like wheat and barley. And in fact, in the monocots, there isn't an FLC, but there's a slightly different gene. Similar inputs, but it's a slightly different gene that's evolved to do the same kind of thing. When they look at that, yes, there's a direct effect of, the influence of the regulation of that on yield. And in the brassicas, it's more the sort of vegetable bits you're harvesting, so it's a little harder to do the yield analysis. I guess in the brassica, oilseed rape, it's certainly true.
Well, Caroline, I would like to thank you for a truly stimulating talk. You've given us a lovely introduction, but actually more than an introduction to this wonderful subject. And of all the weeks to do this, because for those of you who don't know, it's the 100th anniversary of the Genetics Society, and we were at the John Innes Centre on Tuesday celebrating this, and it is an amazing field. And I was extremely struck by your closing remark, really, about where the field has come and our knowledge base, but also in terms of there is amazing stuff that we can do and is being funded to go and put things on, a comet, which is incredible engineering. But when it comes to thinking about the issues of today, such as the implications for agriculture and climate change, there are some significant challenges that we have as a society, and this research is truly, truly important for actually the safeguarding food supplies, etc., for the future of the planet. So if you would join me in thanking Caroline in the most normal way. Thank you so much indeed.